In a bustling genetics laboratory in Japan, a team of dedicated scientists stands at the forefront of a remarkable breakthrough. Using the powerful tool of CRISPR gene editing, they have achieved what was once thought impossible: the removal of the extra chromosome 21 from human cells, a genetic anomaly that underlies Down syndrome. This development marks a momentous stride in biomedical research, offering a glimpse into a future where the consequences of chromosomal disorders might be mitigated or even reversed. Down syndrome, also known as trisomy 21, arises when an individual inherits an extra copy of chromosome 21. This additional genetic material disrupts normal development, leading to intellectual disabilities, distinctive facial features, and a range of health concerns. For decades, scientists have sought ways to understand and possibly correct the genetic basis of this condition, but the complexity of entire chromosomes has presented formidable challenges. The Japanese researchers approached the task with precision and caution, harnessing the capabilities of CRISPR, a revolutionary gene editing technology that allows for targeted changes in DNA. They devised a method to selectively remove the superfluous chromosome from cultured human cells. The procedure required not only technical expertise, but also a deep understanding of cellular biology. By guiding the CRISPR machinery to the precise location of the extra chromosome, they were able to excise it while preserving the integrity of the remaining genetic material. The results, observed under the scrutinizing lens of the laboratory, were striking. The corrected cells, now bearing the standard complement of chromosomes, began to behave differently. Freed from the burden of extra genetic instructions, these cells grew more rapidly, mirroring the behavior of typical human cells. Their gene activity normalized, no longer exhibiting the erratic patterns seen in trisomic cells. Importantly, the cells showed reduced stress responses, a sign that the correction had alleviated some of the cellular turmoil caused by chromosome imbalance. These findings are still in their infancy, limited to laboratory cultures and far from clinical application. Yet the implications are profound. For the first time, scientists have demonstrated that it is possible to address the root cause of Down syndrome at the cellular level, offering hope for therapeutic strategies that target the condition's genetic foundation. The prospect of developing treatments that could one day benefit individuals with Down syndrome, perhaps by correcting cells in specific tissues or organs, now feels a step closer to reality. However, the journey ahead is long and complex. Removing an entire chromosome from living cells is an intricate process, and translating this technique from cultured cells to a living human being will require overcoming significant biological and ethical hurdles. Researchers must ensure that such interventions are safe, effective, and do not introduce unintended consequences. The ethical considerations are equally weighty, as society grapples with the implications of altering the human genome. Nevertheless, the breakthrough in Japan underscores the transformative potential of modern genetics. It highlights the ingenuity and persistence of scientists who, through careful experimentation and relentless curiosity, push the boundaries of what is possible. This achievement serves as a beacon for future research, inspiring teams around the world to explore new ways of treating, and perhaps one day curing, genetic disorders at their source. As the story unfolds, the world watches with anticipation. The work in Japan may be only the first chapter in a much larger saga, one where the mysteries of the human genome are gradually unlocked, and the promise of gene editing technology is realized for the benefit of all. The path forward may be uncertain, but the destination, a future where genetic conditions can be understood, managed, and perhaps even overcome, is now visible on the horizon.